and I am going to take a biology class. So in our continuous series of the classes, we are talking about biotechnology and its application. In the last class, we have discussed about gene therapy and molecular diagnosis, and in that we discussed that how gene therapy is going to be useful in the removal of the genetic disorders, yes, which is present in the organisms by the birth. Then molecular diagnosis at the molecular level, the micro percentage, if the microbes or positive agents are present in the person or in a host causing infection, that can be determined before giving the symptoms. Yes? So lots of things are there that we have discussed. Now in today's class we are going to discuss about transgenic animals. Okay, we are going to discuss about transgenic animals and their use. Now actually transgenic animals are not present naturally. Okay, transgenic animals are not present naturally. Okay, they are not present naturally, but actually they are the result of genetic engineering. They are the result of genetic engineering. Okay, they are the result of genetic engineering, or we can say with the help of recombinant DNA technology, they can be produced. Now what we do in recombinant DNA technology, that in transgenic animals, or we can say when a desired gene, okay, when a desired gene from one organism, is collected or isolated, okay, collected and integrate into another host, okay, with the help of vector. That desired gene gets incorporate into the genome of host. Okay, so thus your transgenic animals get developed, but they are actually the genetically modified organisms. What we do, suppose we have chosen a desired characters of any particular organism, and the gene which is responsible for that characters, we isolate them, okay, with the help of different methods that we have discussed in earlier classes. Now what we do, that isolated gene, okay, of desired character is transferred in a cloning vectors or in a vectors, okay, and then that vector that needs to be feasible, or that can easily get transported into the particular host. After that, what happens, the gene which is carried by that particular vectors is transfer that into the genetic material of the host. So in this way, now the organism in which we have transported or we have integrated this desired gene going to show its particular characters and expression. So now the organism which is constructed or developed is known as transgenic animals. Okay, that is known as transgenic animals. They are not naturally present, but yes, they are the result of genetic engineering or biotechnology. Okay, so why we have moved in such kind of steps, or what are the advantages that we can obtain from the transgenic animal. So, thing is that before going to discuss in detail, we all know that nowadays there are different types of researches are going on in the field, scientists in science, okay, and to know the effect of that research or the result of that invention on the human beings or on the mankind and other animals, it need to be tested. Correct? Suppose if we have developed any vaccines, any medicines, or any other chemical product, so check whether that product is feasible for the humankind or not, what are the consequences, okay, so we need to apply that in some organisms. So actually the transgenic animals, okay, the transgenic animals are acting as model organism. They are acting model organism, and because of the modification, because of the changes in their gene, or we can say genome, okay, so the response to that particular chemicals or whatever the test that you want to perform may vary. So there are some special features, okay, that are related to transgenic animals. We will going to discuss one by one. Okay, what are those features? Okay. Now, what are those features that we can see is to study normal physiology and development. To study normal to study normal physiology and development. Now, one more thing, the transgenic animal that we select, or the model organism that we select, or the organism on which we want to perform the experiment, we need to be careful that we can easily control them. Okay? It is very much necessary that we are able to control them, because there are chances, because the disadvantage with the genetic engineering or transgenic organism is that whatever the changes that we are doing with their genome, we don't know that what may be the result. Yes, so we should be very much careful that if there should be any negative result, okay, that can be easily controlled. So first thing, to study normal physiology, and development. Now, in case of human or other organisms, nowadays we have dissected, we have researched each and every part of the body, that what kind of hormones, what kind of enzymes are secreting, and we also come to know that if there is any defect in a particular organs, what are the other consequences going to be occur for the growth and development. So transgenic animals are also playing very much important role to study the normal physiology, any living organism or human beings. Okay, because see, we are also mammals and there are many other mammals. You have seen that in recent, okay, that we will discuss in later. Okay, so but normal physiology and growth development of an organism step by step can be easily studied with the help of transgenic organism. Okay, with the help of transgenic animal. Now, study of disease. Second thing that we can see, study of disease. As I told you, the transgenic animals are going to act as model organisms. So what we can do, we can select them and we can infect them with a particular causative agent. Yes, and we can study the symptoms that is going to appear in that organism, and also we can use the medicine or whatever the tests that we need to diagnose with the disease. We can perform on them, and depending upon the results that we will see, we can select another transgenic animals. In this way, we are going to study the different types of disease. Okay, there are lots of like carcinogenic disease are there, tumor-related things are there, other infectious disease are there. So that disease, that infectious agents going to cause what kind of changes in the body, that can be studied with the help of transgenic animals. Okay, and after that we can also check the what is the response by that organism against the medicine that we are using for that particular disease. So transgenic animals also playing important role in the study of the what? In the study of the disease. Okay, how come this is going to be helped? See, in transgenic animals, the thing is that we are doing with the gene, change in the gene and genomes, right? So in normal condition, what our body do respond, suppose we are suffering from the disease, so the DNA, okay, which is having the gene and which is having the different kinds of information for the antibiotic formation or fighting cells and such kind of thing. So in what way our immune system or how the gene going to express what kind of protein, all those things are going to be, all those things can be studied, because every time we are not going to use and to take disease. There are many things that our body itself fight against many of the disease. So all those things can be studied with the help of the transgenic animals. Okay. Now the next thing we can say biological product. Okay, biological product. This is a very much important point that we are discussing, because biological product, it means that the things that we are obtaining is from the living organism, directly. Because see, there are two things. One thing, that if we are going to make any product in vitro condition, okay, in vitro condition like by artificial method, that is going to be costly. There is chances of mismatch and lots of problems may be there. Okay, say in the artificial condition, if you are going to prepare insulin, okay, that is going to be more costly and typical. But what we can do, suppose we select a particular organisms, okay, host organism, and we isolate the gene which is responsible for the our desired product. Suppose we have seen in the case of yeast, in the case of many bacteria, yes, there are such kind of things in which we can insert our desired gene, and the product that we want, we can easily or naturally we can obtain that from them, instead of creating that product inside the lab or in vitro condition. So first of all, that is going to be easy, it is going to be grow easily. Yes, suppose a bacterial culture, no needed very much complex things. Okay, simple culture media you provide, and your recombinant molecules or the gene that you have transferred with the help of plasmid in the bacteria, and the bacteria are going to grow, divide, and you will finally get the product. Okay. With the biological product, we have discussed about phenylketonuria. Yes, phenylketonuria. If there is a deficiency with this gene, okay, that can be obtained. This can be obtained by the in vitro culture, means with the help of the organism in which the gene can be transferred, and this product can be going to be obtained. In the case of insulin, yes, we know that in our body the insulin is actually in the is connected with C peptide. Okay, insulin peptide, polypeptides, and A and B, which are connected with C peptide. So, but in mature case, in mature insulin, you will not obtain that C peptide. Correct. So, what we have did, instead of creating them in the lab, we have transferred the gene of peptide A and peptide B into the E. coli, okay, with the help of plasmid, and simply what we have to do, we have to only join them with the disulfide. So these things are going to be beneficial, and easily we can obtain the product which is very much complicated to create inside the lab. Okay. Now the next thing, cases, or many times we found the cases that after the birth, the mother is not able to produce good amount of the milk to fulfill the demand of the baby. So in that case, the simple cow milk which we use is not sufficient for the immunity of the baby, okay, for the immunity and for the fulfillment of the nutritional demand of the baby. In that case, now what happened, the scientists have created cow that is rosy, okay. In that they have given some modification, incorporated the gene which is equivalent to the milk of mother. Like in human, okay, in human males or in the human females, the milk which is present for the growth development, okay, the protein which is there in the milk for the baby, it is calculated or it is estimated that near about 2.4 gram, okay, in a liter, that particular protein is present which is produced by the human mother or female, okay. So such kind of things now, like they are giving a very much beneficial role for the human beings, okay, to come up with the lots of problems that they are facing in their life. So in this way, transgenic animals are used for the, there are lots of examples. In a simple paragraph, we are not able to complete this, okay. If you people want, you can go on net and search that lots of, biological products we are getting nowadays, wine industries, bakeries, then the fungi, in the case of suppose every person are not possible to buy the vitamins, tablets and everything from the market. So there is easy source for scientists, vitamin A rich rice, potato, in which what they have incorporated that kind of genes which are going to be increase the nutritional value of that particular food product. So by the simple form, a person going to consume the food, it is going to fulfill the demand of that particular vitamins, minerals and nutrient that needed for the growth and development of their body. So in this way there are lots of things are there, okay. It's not like one food product. In the case of medicine, in the case of food, in the case of ointment or medicinal values, lots of things are there. Now the very much important next point that we are going to discuss is vaccine safety, okay. Vaccine safety. Okay, now what do you mean by vaccine? See, in the case of vaccine, actually the positive agent is incorporated or is injected into the host body, correct? But condition is that they are attenuated form. They are attenuated form, or the positive agent, like in polio vaccine, what we add, what we inject, actually we inject the polio virus itself in the body. But that is attenuated or disarmed form, okay, disarmed, okay, disarmed form or attenuated form, okay. You can say pathogen injected. So what happens? The body immune system going to respond against that particular positive agent and produce the antibody, right? This is, so it's not like simply we prepare a vaccine and we get inserted, okay? Though in the case of corona, so it's so pandemic condition also, okay, it took near about more than 1 year or 2 years time, near about, due to the final development of the that vaccine. But before, or in normal conditions, what happened, it took more than 10 years time, okay. Nowadays there are lots of technology which make our process fast, but initially there were lots, it was very time taking process. And second thing, the transgenic animals are a very much important and good source for us to check the effect of the vaccine, because simply we cannot use human as a model in the initial time, okay, in the initial period of the trial or the testing of the vaccine. So we use from mice, then rhesus monkey, and lots of there are transgenic animals are there that we use to check the safety of our vaccine. check the safety of our vaccine, that the vaccine that we have developed is suitable for the human beings or not, whether it may going to cause some other severe problem, does it getting mutated after injection. So lots of things are there, criteria that we need to check the vaccine before to release in the market. So transgenic animals are playing very much important role in checking the safety of the vaccines that we have developed by the research. Okay, and in the vaccine actually we are injecting the positive agent, but which is inattenuated and disarmed form. Okay, now so vaccine safety. Now the next thing is chemical safety test also. Like say the medicine or the drugs or just the vaccine test safety test, there is also chemical safety test. Okay. So I told you, like drug and medicine test, vaccine test, there is chemical safety test. Nowadays, we are using lots of cosmetics, ointments or other products on our skin, on our face, in our eyes. So before using that, they need to be tested that whether they are going to be cause any allergy or any side effects. So that purpose also these transgenic animals are used. The advantages with transgenic animal is that they show quick response. Okay, they show quick response against the chemical. Okay, so because of the genetic modification, they show quick response regarding or against the particular chemicals that we are using. Okay, and depending on the symptoms or the result, we can change them, we can dilute them, we can concentrate them, or we can study other consequences also. So actually, the transgenic animals are giving us lots of facility, and we can say that actually they are now a source for us to come down or to make settle our curiosity regarding different kinds of development that we have discussed and that we have developed. But there are also ethical issues that whatever, there is life in them also. So the test or the experiment that we are using for them should not get turned into cruelty. It should be not like going to develop a sense of feeling of cruelty. There are rules and regulations, and there are ethical issues with them also. Okay, that in what form and how can you are going to be use them, and it should be not in such a way that which may be dangerous for the human generation or for the environment, for the biodiversity or for the other organism. Because suppose that our experiment or our transgenic animal that we have prepared, any reason if it get exposed to the environment, okay, and release themselves into the environment and get mutated and do any severe cause, that will be very much serious problem for us to control them, right? So there are lots of rules and regulations that we will discuss them also. Okay, so what we have discussed in the case of transgenic animals, that to study normal physiology and development of organism, the body, at which stage organs are formed, which organs have what kind of function, what kinds of secretion they have, and related with them, the genes responsible for them, that study of disease. There are different diseases nowadays, and we have developed different types of and lots of medicine against that disease. So that also with the help of transgenic animals, biological product nowadays, the biological product that we are developing like chemical medicines, directly we are obtaining from the organism, but just simply by transferring the gene into them, and naturally we are producing the required product, okay, instead of artificial, which is very much costly. Okay, Then vaccine safety. Nowadays we have lots of vaccines for serious type of disease, right? And that vaccine are tested and get assured with the help of this transgenic animal. So, and in the chemical safety, same like vaccine test, there are chemical safety tests also. Okay, so transgenic animals we can say is going to be, they are actually acting as a boon, and in coming future also they are going to be like boon for the humankind, because the research, the development that we are doing, they are giving us a very good platform for the success, for the trial, for the test, to check the result of our experiment. Okay, hope you people have understood this topic and the importance of transgenic animals. Just a minute. Okay, so with this topic we are going to finish today's class. Thank you and have a nice day.